Hello, everyone. Thank you for tuning in. Uh, of course, we all wish we were together in Montreal, but I'm grateful for the organizers putting together this virtual conference so that we can at least connect through science this way. What I will try to do in the next 45 minutes is to tell you about our work on population neuroscience of the growing brain. But I will try to do that in the context of big data, how how we got here and how we should we may want to think about this kind of research in this context. Here is an outline of my lecture. I'd like to start in, if you like, give you a little bit of personal and historic perspective. So I will start by talking about power of observation, because at the end of the day, science is about observing and experimenting. And then I will give an illustration of what kind of big data or forest of data we are dealing with in terms of its breadth, depth, and sample size. And then the most part, the main part of the lecture will be really about looking for those trees in the forest of data, trees that explain a phenomenon. And that's where I will focus on brain development and maturation. So let's start with the power of observation. And I'd like to go to someone that most of you know, Professor Brenda Milner, someone who has been living and working in Montreal since the 40s, and coincidentally, someone who survived Spanish flu. She is still active. She hasn't retired, going on 102. She was my mentor when I came to Montreal in 1990, and she was a Talarak lecturer when we organized the conference, Human Brain Mapping Conference in Montreal in 1998. And as many of you, those in particular, those interested in memory know, she is the one who discovered, who described the patient HM and his severe memory deficit that followed excision of bilateral excision of the hippocampus and adjacent structures. And really, it was the initial observation that then led Dr. Milner to embark on the path of trying to understand the brain behavior relationship in the context of memory. And of course, in those days, she went from that single case to a group where she was verifying, fine-tuning, and discovering that initial observation. And of course, without the neuroimaging, the only way to know where the lesion is were drawings like this one, provided by a neurosurgeon following surgery for intractable epilepsy. It was in the 70s when a key discovery for our field took place. And that's really the development of echoplanar imaging, MRI, with Peter Mansfield in Nottingham in the 70s. You can see the scanner there that they built, and the first picture of the brain here. That's, of course, post-mortem. Not a lot of detail in there, of course. And now look at the differences. Over the past, 40 years, we go from an image taken on this 0.1 Tesla scanner to an image that we took in mid-90s. In fact, that's the year when we held the first conference for human brain mapping in Paris, so 25 years ago. An image taken from a volunteer who went into 1.5 Tesla scanner 27 times, and those 27 images were averaged to increase the signal to noise ratio. So we have incredible level of contrast and granularity. And of course, you move forward and you move to higher field scanner, and you can achieve similar, if not better, of course, signal in a single scan. Now, these were, of course, again, single images, but again, the power of group is in averaging them. And there the key advancement that really moved the field forward was the concept of standardized stereotaxic space. And I'm just showing you what happens if you align, in this case, 800 plus adolescent brains within, in this case, the ICBM or MNI 152 template. You get, of course, a good appreciation of the average of the structural properties of the brain. And you can, of course, do that also for brain function. Here I'm showing you a probabilistic map of the brain response to faces based on over 1,000 14-year-old adolescents that we have, data that we have acquired in the context of Imagen. And just few observations here. When the individuals, the kids were looking at ambiguous faces, you see that there is a number of regions that showed a robust response to faces across the population, more so for the right hemisphere compared with the left hemisphere. And the response to ambiguous faces was stronger overall than a response to angry faces. So the brain had to work harder to figure out, or trying to figure out, what those social cues carried by a dynamic face, what those cues are about when the face was ambiguous rather than simply angry or happy. Now we can again do even the same thing with lesions. We can map lesions in the standardized space and then ask which regions are critically involved in a particular function or paradigm. And here again, we go back to faces and see that there is a great overlap between the lesion and deficit in facial recognition tests in the right hemisphere, and in particular, and again, in regions that involve posterior temporal and parietal regions and fusiform and other visual regions in the occipital and temporal lobe. So I think that this was a very brief introduction to the idea that you, of course, can start with a single case, powerful observation, and then you build on that as you go on, and you acquire more and more data. With imaging, of course, the richness of those data sets the richness is incredible. So let's move now into population neuroscience. And when I started in working in that 20 years or so ago, really the main motivation was the realization that we are, of course, dealing with a complex organ, the brain. We are dealing with incredible complexity when it comes to genes, and even more complexity when we deal with environment, whether it's social environment, physical environment, what have you. Genes more or less remain the same throughout life in terms of genetic variations. I'm not talking about epigenetics here. But of course, the environment continues to change around us before we are born and then throughout our life. And of course, the genes and environment interact with each other in influencing the brain. So that's why many of us started thinking about large numbers. Of course, geneticists knew that before we got there. And that's where population neuroscience comes in, as a combination of epidemiology, genetics, and neuroscience. Epidemiology and genetics on the side of exposures, if you like, that is the factors that are, if you like, independent variables, and the neuroscience on the side of mechanistic insights, and also, of course, the phenotypes, the outcomes. In the case of most of my research, structural variations or variations in brain structure. Now, let me illustrate the power of this approach on a study that we started almost 20 years ago, the SAGON-AU study. At the time when it began, it was arguably the largest population-based collection with wide range of data. So at the end, we collected data in over 1,000 teenagers, aged 12 to 18, and about 1,000 of their parents. They were siblings. That's why the number of parents is the same, right? So they were close to 500 families. Now, we are now in the process of following up those adolescents, and we have also done some work, phenotyping work on the parents. And very briefly, that was the design of the study. As I said, close to 500 families coming from genetically and environmentally a little bit more homogeneous area of the Saguenay-Lac-Saint-Jean region of Quebec. And we were interested in this particular case in prenatal adversity. And so half of our participants were exposed to maternal smoking during pregnancy. So I'm referring to those as exposed, 211 families. And then we matched by maternal education and school attended to those exposed about 228 families with not exposed adolescents. And of course, as you can imagine, matching for maternal education is very important because that's one of the main differences between mothers who smoke or do not smoke during pregnancy. I won't be getting into the details of what we found. That's all published. I just want to illustrate the flow of a study of this sort. As you can see here on this slide, the data collection really begins with a follow-up phone call to the parents. Then we have a telephone interview, a long home visit where parents and the kids are filling out a number of questionnaires. We are getting blood samples from the parents. Then we go to the school lab and do 6 hours of cognitive testing. Then we had a hospital section that included imaging and cardiometabolic phenotyping. And then we went back to a school where we obtained fasting blood samples in the morning for both proteomics and genomics. Now, obviously, from all those sessions, we get a lot of data. So we have a database that contains all the different instruments. These might be questionnaires, different acquisition sequences with MRI, etc. So over 50 instruments that we describe. And then we go into a particular instrument, for example, measurement of different structural features of a voice box from the MRI images. And then we have the different variables. And of course, in this way, across those 50 different instruments, we have several thousands of variables. And this table, don't worry about the details here, is just showing, listing different variables, in this case, ordered by sex differences in the measurement. Effect sizes high on this side mean males larger than females, and on this side, the opposite, females larger than males. And that work was published a few years ago. And essentially, we see when we look at 66 traits that go across the brain and the body, that 11 of those have large effect size, 13 medium, and then most of them have small effect size when it comes to sex difference. We can then use those, and that's a work that is being carried out in my lab now by Daniel Fosberg, that we can now calculate a composite sex scores across, in this case, 46 brain and body traits, and look at the distribution of those sex scores within each sex, males and females. And of course, by definition, those sex scores that are weighted by the effect sizes that I just showed you are different between the two groups, but of course there is also an overlap. And so there are males that have more, if you like, feminine features of their phenotypes, and there are females that have more masculine features of their phenotypes. And we are now working with this continuum of sex differences within each sex to understand inter-individual variability and on that continuum scale. So let me now get into the brain development and brain maturation. Let's start with some basics. So human brain and in numbers, we see that most of the brain mass is really the cerebral cortex, both gray matter and underlying white matter. We also see that the number of neurons is not the highest in the cerebral cortex, it's the highest in the cerebellum. And we also see that there are differences in the ratio between non-neuronal or glial cells and neurons, and that ratio is very different in the cerebral cortex in favor of glia, and the opposite in favor of neurons in the cerebellum. Now, there are many different features that we can extract from structural images of the human brain, and I will focus on two simplest features in this presentation. And that's surface area or total volume of gray matter and cortical thickness. From developmental perspective, you can see that there is a huge, of course, increased growth of, in this case, total cortical gray area during the first the second 20 weeks of gestation. So we go from 20 weeks at 0 to almost 150% change by birth, and another 150% change by about the age of 2 years. But as you can see, the overall growth and surface area, right, that's illustrated here, is not changing much after the first 2 years of postnatal life. And that's very important, and I'll come back to that. So let's start with the surface area that we can, of course, extract using, for example, FreeSurfer. And in the first study that I want to tell you about, we were interested in the molecular architecture of that cortical growth, the tangential cortical growth reflected in the surface area. And we set up that research in the context of the CHARGE consortium, set up as a consortium of cohorts for heart and aging research in genetic epidemiology. And you can see the list of the founding cohorts right here on this slide. And with a postdoc, Jean Shin, what we have decided to do is to do meta-GWAS in the CHARGE consortium using MR images from and genetic information from over 20,000 participants. Remember, these are adult participants. Now, many of you know that with FreeSurfer, you can segment the cortical regions based on different atlases. One of the most common segmentations is Desikan-Kiliani one, 34 different cortical regions per hemisphere. And the first thing that we did was to ask how those different cortical regions, when it comes to their surface area, hang together. So we did principal component analysis of the surface area of those 34 regions. And acquired, in this case, I'm showing you PC1, which is basically reflecting the global growth of the cortical surface area during those first few years of life. And then we use that variable, the PC1, as input into our GWAS analyses. And the meta-GWAS revealed a number of highly significant loci, in particular here, this one on chromosome 17. You can go to the paper that was just published a few months ago for details, but I don't want to dwell on PC1 here. I want to move into PC2, which is really particularly interesting. You can see that the PC2 really brings together primary visual cortex and adjacent secondary visual cortical areas. So something very different must be happening with the growth of regional growth of this part of the cerebral cortex. And when we use PC2 as our input variable into the GWAS, we reveal a number again of significant loci, genome-wide significant loci, with one particularly striking on chromosome 14 that contains a gene called disheveled associated activator of morphogenesis 1. And this gene is very important in the planar cell polarity pathway. And that's a pathway that is critical. Here you see the down one, is critical for cell migration. Now, remember, most of the scans that went into this analysis are scans of adults. So how come that we are pulling out a gene that apparently is related to development? And when it comes to the expression of this gene in the cerebral cortex, as you can see on this slide, the peak of its expression is actually in mid-gestation. And then by the time we are born, the expression levels of this gene are virtually zero. Now, clearly, and that goes back to my point about the brain growth and the growth of surface area being finished more or less by the age of 2, that phenotype is as if frozen in time. So if I look at your surface area or my surface area today, it would be virtually the same as it was, in my case, 57 years ago. And that gives us incredible power to study development. Now, just a few points here. In fact, we know, and that's the radial unit hypothesis proposed by Pashkorakic, that essentially it's the number of ontogenetic columns that is determining surface area. And the migration, of course, then from the phase of symmetric divisions around the ventricular zone, this migration, of course, needs signals of cell polarity, and that's where down 1 comes in. And it appears so that there is, of course, also very important input, that's thalamic radiation, into the subplate zone that may help with the migration process. We know from other studies that, in fact, if you eliminate that input by, for example, removing the eyes of the fetus in non-human primates, the size of the visual cortex is reduced dramatically. So, of course, for me, the final point about this study is what does it mean functionally? And this illustration from a wonderful review paper by de Souza and Proul is showing a very basic idea. If you have a larger visual cortex with more neurons, then it's as if you had a TV screen with higher fidelity. You have of course, many more units to represent visual fields. So you are representing more details. And so we do know across species that visual acuity goes up as a log of volume of the primary visual cortex. The other interesting thing that comes down to that reflects that increased fidelity is the fact that visual, for example, size illusions like the Ebbinghaus here, that is the influence of the surround on you judging the size of the central stimulus, which is of course the same size for those two examples here, but it appears smaller here compared with here because of the influence of the surround. That kind of illusion is weaker in a species with larger cerebral cortex. So the final thing that I want to tell you about is how we try to link or explore the possibility that there is a brain-behavior relationship in this particular finding. So is it the case that individuals with larger visual cortex have better vision, if you like? And we, of course, didn't have data in those 20,000 participants on visual illusion or visual acuity or anything of that sort. And of course, those data would have been anyway confounded by many other things. So what we instead decided to do is to go and take advantage of a knowledge of aptitudes that are important for certain occupations. And we did that in the UK Biobank, and it made source of data, as many of you know, that includes many different variables, including genetic information in almost 500,000 of participants, with the plan 100,000 of them having MRI. MRIs. Now, at the point when we were running the study, we had access to about 20,000 of those MRIs. And what we did, as I mentioned, we did go into the occupational aptitudes, and we were particularly interested in one that is called form perception. And in the dictionary of occupational titles, that is the basis for assessing aptitudes across 20,000 different occupations. Form perception is defined as the ability to perceive pertinent details in object or in pictorial or graphical material. So clearly, we would expect that occupations that require higher aptitude in terms of form perception, that individuals who hold these occupations, and here are a few examples of those occupations, that individuals who hold those occupations are perhaps would benefit from having larger visual cortex. And that's what we tested and confirmed. Of course, the amount of variance explained in occupation that the individuals held and the form perception derived from that associated with those occupations and the size of visual cortex was tiny. But it was really a matter of proof of principle that we can use, in fact, data as coarse as occupation to get at some very interesting biological detail. And that's all in the paper that I mentioned to you, with Jin Shin being the first author. So let me now quickly move into the second measure. And the second measure that we often work with is cortical thickness. Unlike surface area, as you can see on this slide, cortical thickness continues to change throughout life. There is a lot of change at the beginning during adolescence, then more or less plateau in middle age, and then by about in the mid-50s, it starts going down a little bit as well. So much more dynamics. Now, the example that I want to give you from the work on cortical thickness comes from Enigma, another large consortium many of you are familiar with, that brings together a large number of investigators across many different countries. And the question that my PhD student Yash Patel and I asked is, what are the similarity and differences in cortical thickness in different psychiatric disorders, and what would be the neurobiological underpinning of those differences? So again, we go to those 34 cortical regions, and now we analyze data from 6 different disorders that are listed here. We run, and the data come from about 148 cohorts that we brought together and got the same script that each of the cohorts ran for us, so that we could then meta-analyze the beta coefficients that told us essentially what is the group difference between each of those disorders. So these are cases and corresponding controls. Right. So now what we see plotted here is that mean difference in cortical thickness across the 34 regions for the 6 disorders. And as you can see here, we have several thousand of cases and controls for each of those disorders. Now, just looking at it qualitatively, you see that there are many similarities in this profile of group differences in cortical thickness across the 6 disorders. And we can, of course, evaluate it statistically by cross-correlation of those profiles across the 6 disorders. So here you see, for example, that those cortical profiles of group differences coming from schizophrenia are very similar with those coming from autism, bipolar, and to some extent also major depression and OCD. Now, of course, we can then compare this structural correlation or the structural similarity across those different disorders with genetic similarity, genetic correlation across the same disorders. And these data are coming from Brainstorm Consortium. And you can see again, and that's now an established wisdom, that there is a lot of genetic similarity, genetic correlation across different psychiatric disorders. Now, having those two matrices, we can ask how they relate to each other. So in other words, are there similarities? Is there a relationship between the structural similarity and the genetic similarity? And that's what you see here. And the answer is yes. We explain about 27% variance in cross-disorder structural correlation by cross-disorder genetic correlation. So that was the first set of findings from this effort. But as I said, we were interested in the neurobiology underlying those group differences. And I have to take you on a little detour here to explain to you how we can get at that level. Because of course, from MRI, we cannot get as specific as to know how many pyramidal cells we have, how many interneurons, or how many different classes of glial cells. So to get some sort of proxy of that, we developed a technique that we call virtual histology, essentially taking advantage of single cell RNA work that was done in the mouse cerebral cortex, and that identified genetic markers of 9 different cell types. You see the cell types here. And this is the number of genes that were specific to different types of pyramidal cells and the other cell types. Now we are going to use Alan Human Brain Atlas to map profiles in gene expression to profiles in group differences, and we had to filter out a lot of genes that show inconsistent profiles. So once we do that, we can correlate in space profiles of gene expression for each of the cell types with the profiles of those group differences. And as you can see here, across all those 6 disorders, the strongest relationship is that with the profiles of gene markers of one type of pyramidal cells, CA1 pyramidal cells. And the directionality is such that regions with more expression of this cell type show bigger difference between patients and controls. Now, we focus on this particular component, and we wanted to extract now this commonality, this common element in those interregional profiles of group differences. So we ran principal component analysis on those profiles, identified the first principal component that explains about 40% of variance or so, or 50% of variance. And then we again rerun the virtual histology on this particular component, and you see, of course, the pyramidal cells again come out of that analysis. So what they did next, and that's the last part of this story, is we want to extract all genes that are related to this PC1 CA1 pyramidal cell component. So all those genes, and we did that by co-expressing those genes with all genes that have been assessed across those 5 different gene expression databases. And when we did that, we essentially uncovered 2 clusters of genes, clusters based on their developmental profile, now coming from BrainSpan database of gene expression in the developing brain, with data coming from brains as young as 8 post-conception weeks. So all these are prenatal, and all these on this side are postnatal. And as you can see, that pretty much all co-expressed genes fall into one or the other category. Either they are highly expressed prenatally and then their expression drops postnatally, or vice versa. They are not as much expressed prenatally and their expression is high postnatally. when you look, you use gene ontology analysis of those two different types of genes, those two sets, the prenatal genes and postnatal genes, you see very clearly that those prenatal genes are highly, I mean, the terms that are highly enriched are terms that have to do with development, axon development, axonogenesis, regulation of neuron, projection development, etc., dendrite development. On the other hand, the postnatal genes are very much enriched in processes that have to do with neurotransmission, but also regulation of synaptic plasticity. So the bottom line, the synthesis of this work, what this is telling us, that the group differences that we see, at least the ones that are similar across different disorders, that those group differences may arise for two different reasons. One, that is a reason that has to do with prenatal development, and the other one that may have to do with functioning in the postnatal development, functioning of the brain in postnatal development, and involve, of course, processes of experience-related plasticity. So let me finish up with just a couple of slides about the data coming from yet another exciting data set, and that's the ABCD study. As you know, that's a unique data set that is now in, I believe, second wave of data collection with rich phenotyping of about 11,000 children across the US. And what we decided to do, and that's the work of my students, again, Yash Patel and Nadine Parker, we focus on general psychopathology that can be captured with this by factor confirmatory factor analysis, in this case of CBCL data of 112 items that are coming, of course, from the ABCD study. And you can extract what is called P factor, which is essentially a factor that is loading on most of the different types of symptoms. Then you can have specific factors such as internalizing, externalizing, that are riding on top of that general psychopathology factor. And so let me just show you what we see when we now correlate the loading of the P factor across individuals with those two features of the cerebral cortex across the 34 regions. So as you can see, for the surface area, the higher psychopathology, general psychopathology, higher values of P factor are associated robustly across all regions with smaller surface area of the cortex. And that is highly robust, and we'll be following up these findings with an equivalent of virtual histology that we are working on. On the other hand, you may be surprised to see that when it comes to the thickness in those individuals, there are very few regions that show a difference. It's pretty much flat. And that's very different from what I just told you about patients who were diagnosed with different disorder. And that's the key difference. Here we have essentially general population sample of children, so two key issues, general population and children, whereas in the Enigma study, we have cases and controls with cases having clinically diagnosed psychiatric disorders. So I think that that's the main reason why we don't see anything here, and we saw robust differences in the adults. So to sum up, what I was trying to convey in my lecture is that the big data is not happening in the vacuum. It's not an invention of today. The big data is really taking benefit of standing on the shoulders of giants and standing on the shoulders of observation, of neuropsychology, of cognitive neuroscience, and of course the other disciplines, genetics and epidemiology. And it's that kind of informed approach that I'm sure will be extremely powerful when using big data to ask and answer new questions. And of course, the population neuroscience approach has one particular advantage, and that is that it brings together different levels of analysis: the organ, neural systems, cells, molecules. And today I did not speak about social level, but of course, we and others have been bringing that level to this hierarchy as well. And so when you are able to look at the brain in at the same time across those different levels, you've really learned a lot. And I'll stop here. Thank you for staying on, and I will here acknowledge all my colleagues, trainees, and collaborators throughout the world. Thank you very much.